Hello, this is going to be a recording in case something happens to the June 18th presentation and we need to back up. I'm going to have this ready. It's the BiomeFx shotgun metagenomic sequencing presentation. And this quote by Karen: "There's a real need in the functional medicine industry for a stool test that is accurate, reliable, focused on key functions, useful with therapeutic recommendations, and easy to understand." So today I would like to share three things with you. One, why we chose to offer stool testing, what sets us apart from other tests in the market, and review sample report and ordering information. Effectiveness. We needed to create a more effective tool than what was in the industry. Functionality. Offer a bigger picture of looking at the gut that offers actionable items and lifestyle nutrition supplement tailored to one specific microbiome to fill a need. What is currently offered lacks specificity, accuracy, and actionable steps. Today's stool testing challenges are specificity and sensitivity. Number two, data and analytics. Is this reliable? Number three, interpretation. How does this translate to your patient? Four, lack of functional information. What do you do with the data? Our partner is Cosmos ID. Has the highest degree of accuracy on the market, many competencies in place to prevent bias, range from based on 426 healthy subjects, world's largest database with over 160,000 microbe genomes, functional insights on how the microbe and host interact, third-party testing, most accurate, precise lab, CLIA and GLP certified. So this is great to show you how Cosmos ID is really working across several different industries: pharma, clinical, nutrition, agriculture, consumer, and applied testing. So they're associated with many mainstream companies and government agencies, mainstream companies, pharmaceuticals, etc. The current testing challenges. So 16S is the Amplificon methodology, most popular and current methodology on the market, looking at genus level instead of species level, covers only 1 to 2 hyperavailable regions in the genome, the gene. A given hyperavailable region does not capture all the bacterial lineages equally well. This is the Amplificon. So you can see they will find a certain region and amplify those regions. So it amplifies parts of the gene, artificially amplifies gene, has a high false positive rate, cannot identify virus, fungus, and protists, cannot identify the functional differences, cannot identify antibiotic resistance. So you've got these different sections that are amplified, and you have to guess on which organism it is. And here is the whole genomic sequencing when you can see the entire organism. End-to-end sequencing of DNA sequences, able to analyze the species and strain level, identify bacteria, virus, fungus, and protists, most accurate testing on market with 99% specificity and 95.7% sensitivity. So the BiomeFx difference is that it's the most accurate analysis on the market, higher DNA capture, functional analysis reporting, actionable outcome measures, customized lifestyle and dietary recommendations, faster results, more cost-effective. So we go over the sample report. I like to start at the very end just to get an overview. So the first thing we have to understand is the taxonomy. So all organisms are filed by kingdom, phylum, class, order, family, genus, species, and strain. And here's some examples for humans and for bacteria. So you see humans don't have a strain, but you could perhaps put a social security number there. Whereas the strain of bacteria, it's like a social security number. So this is the gut microbiome composition. And if you were to submit your sample, when you compare it, you can see at a glance, there's some differences in the healthy population. And you'd be able to tell by species how many you have too little of and how many you have too much of. So the phyla is the biggest grouping of bacteria. Good visual to reference all the data on the test. The next level is the family level. And you can see here that it gets more specific and can represent the biological important bacteria that we discussed earlier, and it breaks it down even more. And you can see from the family level that there is a difference. And again, it's a good visual, kind of an overview as you get more specific. And then you start going into the sample test report itself. The first you get is an index. So out of 40, this person here has 36, which is really good. Anything above 30 is excellent. And then you'll see you have an alpha and beta diversity with a complete description here. You have the antibiotic resistome. And this is really not, would you first think that it's pharmaceutical antibiotics, but these are the antibiotics that individual bacteria produce among themselves and how they keep their boundaries. And that will give you a good idea of how resistant the microbiome is to infection. And here we see the gut microbiome index. If we look at it more closely, it's a summary of the overall microbiome health, maximum score of 40, so anything above 30 is excellent, calculated by the alpha diversity, beta diversity, gut resistome index, and the pathobiome, or the pathogen control index. So here we're getting deeper into the descriptions. Alpha diversity, the definition is having to do with species richness, average diversity of species in your gut, low alpha diversity, which suggest antibiotics, stress, toxins, diet, or other factors that cause damage to the microbiome. High alpha diversity is paramount to health, improves resistance and resilience of the microbiome, tied to longevity, chronic conditions, linked to better short-chain fatty acid production and overall increased metabolic production. And beta diversity is the overall composition, variation of species when compared to a healthy population. High beta diversity, you can see here the red dot here, trends in the middle of the cluster. And this is showing compositional similarity or successfully coexisting with the environment. A low beta diversity, the red dot trending away from the cluster, which would be out in any of these four quadrants, is compositional dissimilarity, not able to adapt and coexist within your environment. This section here, the resistome index, this will show whether the ecology is off. Measuring resistance that each bacteria has to one another indicates uniformity, stability, equilibrium within the microbiome. The pathogen control index, the definition said it is control index looking at how capable your microbiome is at controlling pathogens, indicates strength, stability. The score, the higher the number, the more capable your microbiome is able to control pathogens. And here so is an index or showing the sample report with several different pathogens listed. The dysbiosis ratio for Mycetes, which is gram-positive, common species like Clostridium, Faecalibacterium, Bacillus, Lactobacillus, Staphylococcus, associated with type 2 diabetes or obesity. And the Bacteroides is gram-negative, common species Bacteroides, Preventella associated with increased short-chain fatty acids, lean body mass, and better metabolism. The Proteobacteria, the most pathogenic phylum, contains species like Shigella, Klebsiella, and E. coli. The Actinobacteria, Bifidobacteria is part of this phylum, helps balance proteobacteria and combat parasites and fungal overgrowths. And here, a complete description that you want to read through before you do your consultation with your client. Now, the biologically important bacteria in your gut, the different categories are the estrobolome, the sulfate reduction, and the methogens, or the methanogens. Functionally, keystone strains would include the Akkermansia musciphylla, the Faecalibacteria perniciae, and the Bifido species. What's important about the Bifido, or the Akkermansia, is that it's the primary mucin degrader, promotes mucosal health and mucus production, low levels associated with obesity, metabolic dysfunction, and intestinal permeability, and can be stimulated by fasting. The important points about the Faecalibacteria perniciae is that it's an important keystone species in the Clostridia class, as well as the major butyrate producer. Promotes anti-inflammatory processes and mucosal homeostasis. Reduces levels that have been associated with a wide range of colonic inflammatory and autoimmune diseases. Now we have many resources available, including a complete set of videos, which will walk you through each section of this report. And I highly recommend going through those videos, and I will send you the links. And here's some points on the ordering, cost, delivery time, and consults. So once you order, you will order 4 kits at a time, but you don't pay until you register the kits. When you pay, it's $299. The turnaround time is 4 to 6 weeks, and then you will sign up for a consult to go over and review the test. And we have Lacey and Dr. Sam Boltz, who are the individuals that go over the test with you. Okay, and then we already reviewed the appendix. So now I'm going to go ahead and stop.